this way. Okay, welcome to the first actual video lecture with audio that is not broken. You may notice that it says AP Biology with Mr. Austin in the top. That is not me. I'm plagiarizing a little bit more. Just hectic start to the year, being out with COVID. Haven't had time to make Unit 1 slides, which I've never actually done lecture on before. So this is a bit new. I promise you they will be my slides eventually. But his are very good, and I didn't want to reinvent the wheel. So we are going to be talking about foundational chemistry, really the basics of chemistry that you should know. You might have forgotten, depending on when you last took chemistry. But let's get into it. So we are going to be going over some general important chemistry concepts, talking about water and its properties, and then acids and bases. And then finally, we're going to talk a little bit about organic chemistry. And there are some self-assessment questions throughout. Answers are at the end of the slideshow. If you want to do them to kind of check your own understanding. So starting with chemistry concepts from the basics, matter. Matter is anything that has mass and takes up space, aka has volume. We have three main states. Yes, there's other weird states, but basically we have three states: solid, liquid, gas. And everything, living and non-living, is made of elements. So what is an element? It's a substance that you cannot break down further by any chemical means into anything simpler. So it's one type of atom, one thing. In living organisms, there are 6 main elements that you should be able to recognize. They are carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur, also known as CHNOPS, which these, 6 elements make up 97% of all the atoms in your body. So far and away, these are the most common things that we are made of, but there are a few others, such as potassium, sodium, calcium, and chlorine, that'll make up the rest, that last 3%, but it's very small compared to those 6. If we look at the elements on Earth, we can kind of see how, kind of where these come from. Why are we made of these? So again, you can see the breakdown of these elements in living things. And if we look at the Earth's atmosphere, we have more oxygen. Basically, we get our oxygen from the atmosphere, obviously. There's also a ton of nitrogen in the atmosphere, but that nitrogen is not actually available for us to use. We breathe it all the time, but we don't actually incorporate it into our body. We just breathe it in, breathe it out. The rest of the elements are found in the Earth's crust, and we'll generally get from plants and eating other things. So back to chemistry, though. We have the atom. Hopefully this is review, but atom is your smallest unit of matter that still has the properties of an element. So they all kind of act the same. They are made up of protons, neutrons, and electrons. Protons and neutrons are in the nucleus. Protons have a positive charge, and neutrons have a neutral charge. They don't have any charge at all. And then orbiting around the nucleus are your electrons, which are negatively charged. And we don't concern ourselves too much with the different types of atomic models. We can go with the Bohr model just fine. Ultimately, what we care about is the number of protons, which tells us what the element is. The number of electrons can vary a little bit, and in biology, it really does. So, how do we get this information? We can read it off of the periodic table. I've never seen them ask you to do this, but it is possible. So you get the name, the atomic number right below the name is the number of protons. We get the symbol, and then there will be a decimal number below the element symbol that is your mass number, which is protons plus neutrons. You usually just round it to the nearest whole number. Most of them are very close to a whole number. There's a few exceptions like chlorine, which is like 35.45, which is not as easily rounded, but we've never calculated atomic mass. Just good thing to know. Isotopes do come up. They are atoms of the same element. So they have the same number of protons, but they have a different number of neutrons. So they have a different mass. And a lot of them are radioactive. So they are unstable and will actually emit particles that we can detect as they break down into smaller atoms. So you've probably heard of these isotopes in the form of carbon dating. Carbon has 3 main isotopes, carbon 12, 13, and 14. And of them, carbon 14 is radioactive and will eventually decay into nitrogen 14. And so you can kind of take a ratio of how much carbon 14 there is compared to the rest, and that tells you roughly how old a sample is if you compare it to a known age sample. And this is going to get into it. So we do a lot of things with radioactive isotopes. They're really useful because it essentially makes atoms or molecules, in our case it's often things like proteins, it makes them essentially like have a flashlight attached to them that we can track and follow it through systems, through bodies. And if they even undergo chemical reactions, even after the reaction, the atoms will still be radioactive. So you can track like an individual atom through chemical reactions with this radioactivity. If you've been to the doctor recently, sometimes when you get like certain scans, they'll actually inject you with a radioactive dye that helps them kind of track how things flow through your body. And it's all based on this idea of a half-life, which is how much time it takes for a radioactive sample to decay so that only half of it is left. So like how long does it take for a 10-gram sample to become 5 grams? Carbon-14 has a half-life of about 5,700 years. And so you basically can, again, figure out how much carbon-14 is left, and then use this half-life idea to figure out how old the sample is. Tracers is what I was talking about with the, if you go to the hospital, you basically attach a radioactive atom to a molecule and then track it and see where it ends up. And so this is actually how we figured out that DNA is the genetic material. We talk more about this later, but they essentially just took viruses, and in 2 different populations of it, in one of them they made the proteins radioactive, and in one of them they made the DNA inside radioactive. And essentially all they did was track that after they infected cells with these viruses, are the cells now radioactive? If protein ended up being the genetic material, then the protein ones would show radioactivity. But since we know that it is DNA, the radioactive DNA was injected into the cells by the viruses, and we then knew that that is the thing that's actually encoding the genetic information, is DNA and not protein. It was an open question for a long time. Okay, compounds. Compounds are 2 or more elements bonded together. Notably, it is not 2 of the same element bonded together. Like oxygen, nitrogen, and fluorine all exist as what's called a diatomic molecule. So it's O2, N2, and F2 naturally. It's still an element because it's the same thing. But atoms, their goal is to get a full valence shell, which is a full, usually 8 electrons. Unless they're very small like hydrogen and helium, then you just need 2. But that's their goal, is to fill up to 8 total electrons. So generally, as long as you are either close to 8 or close to 0 in your outer shell, you're trying to get to that 8 or 0. So nitrogen has 5 valence electrons. It wants to get 3 more. Chlorine has 7. It wants to get 1 more. But for example, something like sodium has 1 valence electron, and so it would rather just get rid of it, because it's easier to get rid of 1 electron than to gain 7 electrons. Bonding types, they do come up. We have ionic bonds and covalent bonds. Ionic bonds are a complete transfer of electrons from one to another, and you get a net charge afterwards. This has to be like your column 1 and 2 with one of the 2 far right columns in the periodic table. And covalent bonds, which are much more common for us in biology, are the ones where you're sharing electrons. So this is something like water. Pretty much anything with carbon in it will all be covalent bonds. And covalent bonds tend to actually be a bit of a stronger bond. Ionic bonds can kind of drift apart from each other because they're just a transferring of electrons. It has to be in water. Whereas covalent bonds, you can't really separate the individual atoms easily. But that sodium molecule there doesn't have to pair up with that chlorine ion. It can pair up with any chlorine ion that is around, and it will. So ionic bonds are pretty easy to break, whereas covalent bonds are not. Ionic compounds generally are salts. They're a positive charge and a negative charge, usually a metal and a nonmetal, so something from the left and something from the right. And when you put them in water, they dissociate, so they split up into their two ions and get surrounded by water, as you can see in the picture there, which is why they dissolve. Covalent compounds, on the other hand, are basically usually what we call molecules. Again, you cannot separate the individual atoms. The water just kind of clusters around them instead, not fully breaking anything up. But individual molecules don't have to stay together. You can still dissolve covalent compounds in water. It's just they don't dissolve as easily. You can't dissolve as much. So sugar, for example, you can dissolve sugar in water. We drink soda all the time, but you can't fit as much sugar as you could salt, for example. You could fit much more salt into the same amount of water than you could sugar because the atoms can break up. So how do you actually break up a covalent compound? Well, you need a chemical reaction. So chemical reactions are needed to break these compounds. You're just changing the bonds, forming new bonds. The classic photosynthesis, where you take CO2 and water and you make oxygen and glucose. Okay, this is one of my favorite topics, and this single slide and the stuff coming up related to it are probably the most important slides in this whole thing, and will come up constantly throughout the year. So we can call molecules either polar or non-polar. And it's really like, are they kind of charged, meaning they're polar, they have poles, like magnets have poles, or do they not have a charge at all? So if they share electrons completely equally, there's no partial charges, they are non-polar molecules. But if there's an uneven sharing of charge, then it is polar. The classic and best example of this is water. Water is an extremely polar molecule because oxygen loves electrons, so it hogs them from those hydrogens, and the result is you get a negative end, which has the oxygen on it, and a positive end where the hydrogens are on it. And so you essentially have a little mini magnet for each water molecule floating around. Nonpolar things are your oils, your fats, lipids. You don't have any partial charges. They're just sharing completely equally, no charge at all. So why does this matter? The biggest reason is because of this principle of like dissolves like. You probably know that oil and water don't mix. Maybe you know that they don't mix because water is polar and oil is nonpolar. But if not, then now you know. It is the polarity that determines if something will dissolve in something else. So the general rule is if it's polar, if it's charged, it will dissolve in polar liquids, usually water. So we see salt easily dissolves in water. But nonpolar molecules are what we call hydrophobic, meaning phobia, they're like afraid of hydro, which is water, so afraid of water. And they repel and don't mix. And so you get those little oil droplets that you see up there. The oil does not want to mix with the water. Whereas if it is polar, we call them hydrophilic. Philic means like to like. And so hydrophilic things like water. They like to mix with water and they dissolve in water. So this really comes up a lot when we do our cell structure unit, which is the very next unit, because it's kind of how our cells control chemical reactions is with this principle. Next, we have molecular shape. Shape is actually hugely important in biology, mainly and especially when it comes to our proteins, because every molecule will have some three-dimensional shape. They don't just all lie flat completely because of that positive and negative repulsion going on between protons and electrons. And so the key thing is that in biology, at least, the shape of a molecule is essential to its function. If two molecules have a similar shape, and shape includes like having similar elements in it, they will have a similar function. So the example you can see on the right there is a natural endorphin, which is going to be like your natural painkiller. Like when you get an adrenaline rush and you don't feel that you actually have a big cut in your leg, you would get natural endorphins released. And then on the right is morphine, which is a synthetic opiate derived from poppy plants, same thing as heroin. And if you look at the boxed area, that structure is remarkably similar. There's a slight difference, right? There's like a little extra oxygen in the morphine there, that red blob. And obviously the rest of the molecule is different, but because that region is very similar, they will both have very similar biological effects because their shape is similar. And so your body generally will react very similarly to morphine as it does these natural endorphins, again, because their shapes are very similar. And they are able, as you can see here, to just bind to the actual proteins in roughly the same way, because it's the similar structure is actually where it attaches to the protein. A lot of the protein structure is not essential to its function. It's just there, but it's not really involved in any of the reactions. So yeah. So interactive, you can answer these questions. Think about them. I'm not going to go through all of these, or we would be here for years, but the answers are at the end of this slideshow, which I will post in Canvas. Okay, I'm going to stop this video and start my second one.